Everyone, today we'll see about immunologic tolerance. What is immunologic tolerance? So, whenever there is any antigen, the specific lymphocyte will get activated and they produce a response in order to eliminate those antigen. Suppose if there is no response, the state of unresponsiveness to the antigen by the lymphocyte or by the specific lymphocyte is termed as immunologic tolerance. In simple word, if a stranger comes, the dog will bark. Here the stranger is antigen and the dog is lymphocyte. Suppose if dog doesn't bark to a stranger, that is a state of unresponsiveness by the dog, then that is termed as immunologic tolerance. So, dog is immunologically tolerant. This is an example to tell what is immunologic tolerance. Now, what is self-tolerance? Self-tolerance refers to lack of immune response to one's own tissue antigen. So, one's own body tissue antigen, if there is tolerance, that is termed as self-tolerance. So, this is what is immunologic tolerance and self-tolerance. But what happens during the maturation process? B and T lymphocyte will acquire certain receptor which will recognize one's own tissue. So, that has to be eliminated. The self-antigen recognition property of B and T cells during their maturation has to be eliminated. Otherwise, what will happen? It results in autoimmune diseases. So, how are these self-reactive antigens are eliminated, or the receptors on T and B lymphocytes are eliminated? This is by these two mechanisms. One is central tolerance, another is peripheral tolerance. Let's see what is this central tolerance. During the maturation of these B and T lymphocyte in the central lymphoid organ, what will happen? The self-reactive B and T lymphocyte are recognized and they are deleted by the process of apoptosis. So, in the central lymphoid organs, these are eliminated. These self-reactive B and T lymphocytes are eliminated by the process of apoptosis, or deletion of these T and B lymphocyte takes place. These, if they recognize self-antigen, they are presented by thymic antigen presenting cells, and thereby they undergo apoptosis. For this apoptosis, or for this elimination process, a protein known as AIRE, autoimmune regulator protein, is essential. Suppose if there is a mutation of AIRE that results in failure of this apoptosis of self-reactive B and T lymphocyte, result in the condition known as autoimmune polyendocrinopathy. But all the T lymphocytes carrying the self-reactive receptors are not eliminated. Some of them, whichever is remaining, they'll be detected and they become what is known as regulatory T cell. Okay, so in the central lymphoid organ, the T cells which are self-reactive are identified. Okay, by the help of AIRE, there is apoptosis or deletion of those. Suppose whichever T lymphocytes which are self-reactive are not destroyed, they become what is known as regulatory T cell. Now, what will happen to self-reactive B cell? Again, they can undergo apoptosis. Otherwise, how they are eliminated? By the process of receptor editing. What is receptor editing? These antigens on the surface of B cells, there is rearrangement takes place. Okay, this rearrangement of antigen causes expression of new receptor which are no longer self-reactive. So, once the rearrangement takes place, they become non-reactive to self-antigen. That is known as receptor editing. So, in the central tolerance, mainly T cells are eliminated by apoptosis, remaining become regulatory T cell. Whereas B cell in the central tolerance, what will happen? Apoptosis, or there is receptor editing. You have to remember something. This central tolerance is always imperfect. That means some reactive B cell and T cell will enter the periphery. So, the autoantigen escape the central tolerance and they escape into the periphery. Now we'll see the other mechanism. Suppose whichever has escaped the central tolerance, how they are eliminated in the periphery. The first step or a first mechanism is anergy. What is anergy? It is functional inactivation of lymphocyte. So what is this functional inactivation of lymphocyte? So whenever there is an antigen, for the T cell to get activated, so the two mechanisms are necessary. One is they have to recognize these antigen on the APC, antigen presenting cell, with the presence of MHC molecule. Okay, so one thing is that T cell has to identify the antigen on APC, presented by APC. Another is by co-stimulatory signal by APC. Now what is anergy? There is absence of co-stimulatory signal by APC, or the receptor on the T cell for this recognition is absent. So the T cell doesn't have that receptor, or this co-stimulatory signal by APCs are eliminated. That process is known as anergy. Thereby what happens? The self-reactive T cell become functionally inactivated. Okay. Next is suppression by regulatory T cell. What is this suppression by regulatory T cell? So I told you in central tolerance, whichever T cell has not eliminated or deleted by the process of apoptosis, they become what is known as regulatory T cell. The best example is the CD4 T cell having more expression of CD25 or alpha chain of interleukin 2 or a transcription factor known as FOXP3. Okay, these are the example of regulatory T cell. Suppose if there is a mutation of this transcription factor FOXP3, then there is a condition known as IPEX, which stands for immune dysregulation, polyendocrinopathy, enteropathy, and which is X-linked condition. So this is the second step in the peripheral tolerance, that is suppression by regulatory T cell. And again, even in the peripheral tolerance, there is a process of deletion by apoptosis. So why this apoptosis takes place? Because of increased expression of pro-apoptotic member of BCL2 family, which result in mitochondrial pathway of apoptosis. Also there is upregulation of receptor of FAS. Suppose if there is a mutation of this FAS receptor, then what will happen? There is a condition known as a mutation of this FAS result in what is known as ALPS, which stands for autoimmune lymphoproliferative syndrome, which is characterized by lymphadenopathy and production of autoantibodies. But you have to remember, some self-antigen are hidden from the immune system. Okay, they are not exposed to blood or lymph. These are known as immune privileged site. As a result, unless they are released into the circulation, these antigen fail to elicit immune response. Okay, but when will this response takes place? When they are exposed to this circulation, blood or lymph, when there is a trauma or any infection. Okay, then they get exposed to this immune response, which result in prolonged tissue inflammation and injury. This is postulated mechanism for post-traumatic orchitis and uveitis. There are self-antigen receptors in the body, but these antigens are hidden in an immune privileged site. Unless and until there is a trauma or there is an inflammation, they get exposed. Then only there is prolonged inflammation or injury. Best example is orchitis or uveitis. So just to summarize, when there is self-reactive T or B cell, you have central tolerance, you have peripheral tolerance. Central tolerance, T cells are eliminated by apoptosis, or which are remaining, which are not eliminated by apoptosis, they become regulatory T cell. Okay, and B cell, how they are eliminated in the central tolerance? Again by apoptosis or receptor editing. Receptor editing is nothing but a rearrangement of the proteins. In peripheral tolerance, how are they eliminated? One is by anergy. Okay, even B cell are eliminated by the process of anergy. And by apoptosis, and you know that regulatory T cell also plays a role by suppression of T regulatory cell. Also, you have to remember one thing: in absence of functionally active T lymphocyte, even if self-reactive B cell are present, they do not cause immune response. So this is overall about immunologic tolerance.